Review of protein synthesis. So as we know, a gene is a segment of DNA that codes for a protein to be made. And we know that RNA has 4 bases, A, U, C, and G, where we replace the thymine from DNA with a uracil. And we also know that RNA is single-stranded, not double. And so all types of RNA are made through transcription, but we mainly talk about messenger RNA in transcription, so mRNA. Transcription, we have RNA polymerase binding to a promoter. So on your gene of your DNA, you'll have a promoter where your RNA polymerase binds to. Promoter just means a certain sequence of nucleotides that it can recognize. It'll bind there, and then RNA polymerase will unzip the DNA just at the certain gene that it needs. So that gene will help it to make just one specific protein. So it'll just unzip at the one little section, as you can see right here, wherever it needs that gene for the protein. And last, the RNA polymerase will build the mRNA strand using the nucleotides. And so it'll have the A, U, C, and G. Again, we know that the T's from the DNA get replaced with U's for the RNA. So next, before we can leave the nucleus, we have to have processing occur before you leave the nucleus, or before you leave the house, is what I compare it to, is leaving the house. First step, you need to remove the introns, so take only what you need before you leave. And then you need to have a cap and a poly A tail. So you're putting on your cap and your boots before you leave the house. And now that you've finished your processing, the mature mRNA can leave to the cytoplasm. And so now in the cytoplasm, you can start your translation. And so for translation, you need an mRNA, so the messenger, it's being sent from the nucleus, the message, to the cytoplasm. You need your rRNA, which makes up your ribosomes, and your tRNA, which is your transfer RNA, to transfer amino acids from the cytoplasm onto your protein chain. And so to start, we have initiation. And initiation, you have all the components that we need assembling and coming together, with methionine starting at the AUG codon. And again, the codon was the 3 base pairs that give you a certain code. We have our lovely little chart that'll tell us what amino acid is used for that code. So methionine is the start AUG. Then we have our elongation. So our methionine tRNA is gone. It leaves after it puts the methionine there. The next one comes in on the next codon. Here we have UAC, which codes for tyrosine. And so once the tyrosine tRNA can come in, it will make a peptide bond here with the methionine, and it'll keep elongating. You'll get a new tRNA come in for the next 3. It'll bring another peptide, a peptide bond will be made, and this chain will keep getting longer and longer, which is why it's called elongation. And last but not least, we have termination. So we had the start codon way over here. We're at the end of the gene that we transcribed. We're at the end of the mRNA. And so now we wind up with a stop codon. And this is where it tells the whole subunit here, tells everything to cut the protein chain off. And the protein will go off to make its own shape. The mRNA, rRNA, and tRNA all go their separate ways. And here is the protein. It leaves and kind of makes its own shape so that it's functional. Don't forget a couple terms. We also had a polyribosome. So if on our mRNA, we had multiple ribosomes, that would be a polyribosome. There's more than one on the same mRNA strand, and they'll all end up making the same protein. So we have polyribosome and codon. So don't forget your codon is the triplet. It's the 3 base pairs that get read together. And we also have the anticodon that would be on the tRNA, so that each tRNA knows which sequence it belongs to. So if UAA comes up, the anticodon would be the opposite bases, so AUU, and that would be the anticodon, so that this tRNA knows when it can come up to the ribosome. And I think that's everything. So that is protein synthesis in a nutshell. You are going from transcription inside the nucleus to processing before you can leave, because you need to mature before you can leave the nucleus. And then you end up with translation, which has initiation, elongation, and termination. And you end up making a protein.